In the past, bovine udder was considered as sterile, and all the bacteria that were identified from raw milk were believed to be contamination of milk. However, recent microbiome studies revealed that the bovine mammary gland contains a rich bacterial community. It indicates bovine udder is a niche for bacteria, and the bovine udder that is mastitis free, and the one that has mastitis forms different bacterial community. Then we can ask questions. What are these commensal bacteria in bovine udder doing when mastitis pathogens invade? Would they give the pathogens some kind of cue for inducing inflammation? Or are they actually good ones that compete with mastitis pathogens? While many researchers are trying to solve these questions, mastitis cases by environmental pathogens, especially mammary pathogen E. coli (MPEG), have been increased significantly in the past few years. However, there are major gaps in our knowledge surrounding this important pathogen. It is still controversial as to whether MPEG is a truly unique E. coli peptide. Thus, marker genes and virulence factors that are unique to MPEG have yet to be defined. In addition, how MPEG interacts with the resident and microbiome of the mobile mammary gland is also not understood. Therefore, the objective of this study is to develop better understanding of MPEG by focusing on two objectives, including genomic characterization of MPEG and investigating how the community structure of the mammary microbiome changes before, during, and after infection in affected quarters by MPEG. The first objective, I have conducted a comparative genomic analysis of larger sets of MPEG and bovine commensal E. coli, which are more than 200 genomes in total by whole genome sequencing and pangenome comparison. As a result, 9 unique genes of MPEG that do not exist in bovine commensal E. coli were identified, which are related to nutrient uptake, putative transporters, zinc metal peptidase, and intimin-like adenosine. For the second objective, I'm going to analyze the raw milk microbiome using 16S RNA amplicon sequencing. These samples include the ones that were produced during the period of mastitis and contain MPEG, and also the ones that were produced by the cows with the history of mastitis before and after the instant of mastitis. This will be able to identify the phenotypes that form healthy microbiome community in bovine udder by comparing microbiome change through the lactation period. I really hope that my research will help to determine bovine udder microbiota that makes the host more either vulnerable or resistant to mastitis by MPEG. It will provide better preventive measures and treatment for the disease, such as antimicrobial treatment decisions. Furthermore, better insight into the development of novel treatments, such as probiotics, can be provided as we identify the potential targets for MPEG.